Welcome back to the second instalment in my new series where I take research concepts and try and integrate them into my previously existing basics videos. I believe this video ties in very nicely with the CSF, blood supply and even neurotransmitter videos that I've previously released. As always, we'll start with the syllabus. Today we're covering the anatomy, the function of the glymphatic system and how sleep influences it, and also its implications in disease. By the end of this video, it's a bit of a bold claim, but I do believe you could walk into any room and discuss the glymphatic system in all its glory. Let's begin. Alright, let's start with some definitions. As I found while doing the research for this topic, I'm going through these PubMed articles on the grind. This is a very definition heavy topic, so it's pretty imperative that I outline a few for you. If we start with perivascular at the top, perivascular refers to the area surrounding a blood vessel. In the context of the brain, these are your cerebral arteries going into your peel arteries and then to your penetrating arterioles. And it's simply that exterior, the region surrounding your blood vessels that supply blood to the brain tissue. Right, next we go down to parenchyma. Excuse if I don't pronounce that properly. But this is very simple, really. It's just the brain parenchyma refers to the functional tissue of the brain. So that's just including your neurons, your glial cells, the extracellular matrix, your glial cells being stuff such as astrocytes, which ties into the next point. In some of the papers you'll probably come across, and if you look through my slides, some of the diagrams, there's two terms, astroglial and astrocytes. These mean the same thing essentially. Astrocytes being a type of glial cell, being found in the CNS, being the brain's spinal cord. They're the most abundant glial cell in the CNS, and they're really crucial. Some people even put it down to when they analysed Einstein's brain, he had a disproportionately high amount of these glial cells, showing they may play a role even in intelligence. But some of their functions literally go from neurotransmitter recycling, blood-brain barrier regulation, all the way to glucose movement. And as I'm about to tell you, they play a crucial role in this glymphatic system. Next we go down to homeostasis. The main basis of this video is really centered around homeostasis. So I just want to really nail in this concept as a key biological concept. And the definition, by definition even, homeostasis refers to the ability of an organism or a system to maintain the internal stability and balance in response to changing external environments. So essentially it's the process by which like a living or alive organism regulates their internal environment to maintain optimal conditions for survival and function despite fluctuations in an external environment. So to put this in context, so I sit in a sauna. If you take my body temperature as 37.5 degrees Celsius and the external temperature of the sauna is 80 degrees upwards, I don't want my body temperature being raised up to 80 degrees Celsius, as my enzymes will be denatured and my functional systems won't be able to work properly. So that's your simple definition of homeostasis. My body's maintaining that internal temperature of 37 degrees by sweating and vasodilation and a couple of other mechanisms. But I just want to drive that in. And bottom for fun is de novo. De novo is a Latin phrase that translates from the beginning or to an anew in English. And it's a term that I saw referred to when talking about lipids, especially cholesterol production in the brain. And by weight of the human body, the brain's only 2%, but it contains 25% of all the cholesterol in the human body. And this isn't the same as circulating cholesterol. All this cholesterol is made de novo, or fresh, in the brain, essentially. And I thought it's just a good little Latin phrase that you can throw into your vocabulary, and just thought it'd just be a bit interesting, really. I want to start with a recap, going over many of the key structures in this diagram seen on the slide, as these are basically structures that will keep coming up and reoccurring in the next diagrams I show you. And if you understand all these key landmarks, the glymphatic system will actually seem very simple, just as long as you have this basic understanding. If we start at the top of the diagram, we have our skull. Beneath it we have our meninges, the meninges being made up of 3 layers: your dura mater, your arachnoid matter layer, and your pia mater layer. The bit between the pia mater and the dura mater is the most important. So if we look at this arachnoid layer, I've drawn on in red these almost spiky projections. It looks like a spider's web. That's how it gets its name, arachnoid. And between these almost spider-like projections called tuberculae, you have vital things such as your arteries and then your arachnoid granulations, and these are the key, arachnoid granulations are the key system by which cerebral spinal fluid is drained, which we'll come on to. Coming in here below the pia mater, we now have your penetrating arterioles. So this is supplying that oxygenated blood to all the cells that need it. And on the other side you have the blue vein being depicted here. Surrounding it, these structures are the astrocytes, and these bits connected to these veins are the astrocyte feet, which are very important in maintaining that blood-brain barrier, and we'll come on to in a bit more detail later on. The other 2 things that I want to briefly mention here is interstitial fluid and cerebral spinal fluid. The interstitial fluid was key in the discovery of the glymphatic system, as within the central nervous system, interstitial fluid was previously thought to move via diffusion. But then recently, I say recently, 2012 is quite a long time ago now, it was observed moving at a much faster rate than possible through the act of diffusion. And by the fact that it was moving faster than it could diffuse, things such as the glymphatic system were hypothesized and then proven. So what is interstitial fluid? It's basically the fluid that surrounds the cells of the brain, those neurons and those glial cells, and it fills the narrow spaces between brain cells, and these narrow spaces are known as interstitial spaces. It contains nutrients, oxygen, and then you've got your ions that are vital for necessary cellular function. So your potassiums, your sodiums, your chloride ions for maintaining concentration gradients and the firing of those action potentials. Then you have your cerebral spinal fluid, which is this clear colourless fluid really that surrounds the brain and the spinal cord, with its primary function being the cushioning when you're subjected to those physical traumas like a rugby tackle or something, and providing buoyancy to support the weight of the brain. It also has a role in facilitating exchange of nutrients and waste products within the brain and the spinal cord. I'm going to outline some of those waste products that are made, as the brain, being a very metabolically demanding organism, creates a lot of these waste products that need to be removed, which I'm going to outline on a future slide. So cerebral spinal fluid mainly made in those chloride plexuses within your ventricles, as I've covered on previous videos, and it's got a constant flow. It's kind of got a beating motion through cilia, kind of like your sinuses really, and you produce about 500 mils of it a day, but your brain can only hold about 150 mils at a time. So if you imagine, it's like a flushing system. The maths doesn't quite add up here when I look at it again, but all the cerebral spinal fluid within your brain is replaced for about roughly 4 times a day. And so this just creates that flushing system, getting rid of all that metabolic waste that can build up within the brain. And the building up of this metabolic waste is what ultimately leads to a lot of pathologies. And this metabolic waste is eventually drained into deep cervical lymph nodes. Is there anything else we need to add? I'd probably like to mention that cerebral spinal fluid does come into contact with this interstitial fluid surrounding the brain cells, and there's a dynamic exchange of substances between the CSF and the ISF being the interstitial fluid. And these include communication or exchange of nutrients, waste products, and neurotransmitters. But when we think of this influx, I want you to think back to that homeostasis. Like, the environment of the brain is very, very controlled, because it needs to be in this almost perfect Goldilocks state for those action potentials to fire, for various functions to happen. And so the cerebral spinal fluid and interstitial fluid and its communication with the blood helps flush away things we don't want and keep that environment in that Goldilocks zone so our brain can function. And obviously, if we move out of this Goldilocks zone, our brain's just not going to work quite as well. And I think that's the key concept. Like, the whole concept of the glymphatic system and its role in disease is based around keeping the brain in this Goldilocks zone. Right, let's continue. Right, we finally get to the actual topic of this video, which is, what is the glymphatic system? Well, the name glymphatic comes from glial cells and lymphatic tissue. As astrocytes, which we mentioned are a type of glial cell, are very prominent in this system. And then, much like the regular lymphatic system, the glymphatic system rids the brain of toxins and waste products, and that's how it gets its name. Right. The definition, if we were to give one, that I would give, is the glymphatic system is a waste clearance system that utilizes unique perivascular channels formed by astroglial cells to promote the efficient elimination of soluble proteins and metabolites from the central nervous system. It's important to add that it's also involved in the movement of non-waste compounds such as glucose, lipids, amino acids, and neurotransmitters. So that's a lot to take in. But if we were to unpack it, just like the definitions we did earlier, we start with it's a unique system of perivascular channels. If you cast your mind back to the beginning, perivascular is that space around, in this case, the arteries. And then what's next? Formed by astroglial cells. So we know astroglial can also be interchangeable with astrocytes. In this case, on this diagram, it's the astrocyte feet. So what is the glymphatic system? It is, if you look at it as a structure, it's that perivascular space, the space outside this artery, and this is bordered by your astrocytic feet. And so that's pretty much all it is, just the space between the artery and those astrocytic feet. And in this space, we can use it as a channel to eliminate those metabolites that we don't want and other substances. One key substance to mention is the movement of lipids, especially cholesterol, made de novo. Another definition from earlier. And these lipids are moved by high-density lipoproteins, low-density lipoproteins within the brain. And a key one of them is a polyprotein E, which transports lipids and also beta amyloid. And if you've done any reading around risk factors for dementias, especially Alzheimer's, you will have come across ApoE4 genes. Pardon me. And these are a risk factor for Alzheimer's, especially if you have 2 copies of them. And this ApoE4 refers to a gene that encodes for your polyprotein E, which transports lipids and amyloid beta within the brain. I just thought you might find that interesting as it ties into the system. Another key thing to mention is when looking at the glymphatic system as a whole, it's important not to separate it from the bigger picture. This was done a lot in papers that seem to only focus on one aspect, like just the beta amyloid, just the tau tangles, just the glymphatic system, and I think it's a bit lost when you're reading about it, especially when you're looking at disease implications. It is all a big system, and maybe it doesn't start with these beta amyloid. Maybe it starts with the glymphatic system, then you move up and you think maybe it starts with cerebral blood flow, which ties in nicely to how does this actually work? What is like the mechanics behind it? So you're with cerebral artery in this case coming through here, your penetrating arterial it pulsates during that cardiac cycle or spontaneous vasomotion, vasovascular and motion movement. And so this pulsation also translates to the perivascular spaces that we've mentioned. But ultimately, this pulsation coming through, the pumping of the heart, is presumed to promote water and cerebral spinal fluid flow along perivascular spaces through aquaporin 4 expressing glial cells. That's a bit of a mouthful, right? So these astrocytic feet making up this channel have aquaporin 4s in them. And a porin is basically a pore or a hole that is going to allow the movement of a substance. In this case, it's water or cerebral spinal fluid. So if you imagine these astrocytic feet have all these holes in, and then when this artery is pulsing, it almost flushes through this water and cerebral spinal fluid. And if you remember earlier when we referred to this metaphor, this flushing metaphor, we're flushing all these waste products out of the brain. This pulsing is allowing that new cerebral spinal fluid, which remember we said gets regenerated 4 times a day. You make 500 mils, your brain can only hold so much. So we're just constantly replenishing it. And it's this flushing motion through these aquaporin channels, facilitated by the pulsing of this cerebral blood flow, that pumps this cerebral spinal fluid and water through this glymphatic system. And it's the pumping and flushing of this waste products, which is really the underlying principle of the glymphatic system not allowing the build-up or specific concentrations of those glucose in the brain to maintain that homeostasis even. Alright, so the vessels that are making up, or these channels that are making up the glymphatic system, all that cerebral spinal fluids getting pumped through, and this cerebral spinal fluid coming into contact with the interstitial fluid that we mentioned earlier. And this interstitial fluid is waste laden. So when they mix together, it basically allows the transport of those waste products and those metabolites into this system, which then gets flushed out. So the next bit is really what's complicated and what's argued about. This mechanism of your aquaporin 4s, the cerebral arteries, and that pulsation and that flushing mechanism, that's all set in stone. But it's what happens next that's often debated in a lot of these papers, and is a bit more complicated really. So now that all these waste products have been flushed out with the cerebral spinal fluid, it enters the dural lymphatic vessels. So if we remembered those meninges, we have our dura mater, our arachnoid mater, and our pia mater. we've then flushed all this stuff back into the space that contains this cerebral spinal fluid, and via the glymphatic system, and then the dural lymphatic vessels transport the fluid into deep cervical lymph nodes via your foramina at the base of your skull. If you think about the anatomy of your spine, the top part's your cervical section. So if you think the lymph nodes coming up through here on your neck, even is your deep cervical area, and then your foramina basically means just hole at the base of your skull, where these are being drained, and then these are going to go into your lymph nodes, and certain compounds are going to be broken down in your liver, etc., etc. But I hope this has given you a good overview of how this system works. Let's move on. Alright, in the previous slides I've mentioned waste products a lot, even saying that the glymphatic system is almost centered around the removal of certain waste products, that being related to the homeostasis and maintenance of those Goldilocks conditions that I've talked about. And some of these waste products that I will come on to mention now hold major weight in the world of Alzheimer's research and other neurological conditions, even things such as head trauma that you can sustain from like a punch in the face or a rugby tackle. And I think if anyone's asked in a lecture what the two usual suspects of waste products that you don't want in the brain are, they'd probably say tau proteins and amyloid beta plaques. And rightfully so, these two things have been vilified in some contexts, but I do believe it is important to know their function in a healthy system and realise that they're actually not innately bad. If we start with tau proteins, these are involved in stabilising microtubules. Microtubules could be likened to a scaffolding of the axon and dendrites that help in structure transport within the neuron. If you cast your mind back to my neurotransmitter video, you've got to think about that retrograde and anterograde trafficking of those certain proteins in neurons, neurotransmitters that are made in the cell soma, that cell body, and then brought down the axon, to the axon terminal. However, in neurodegenerative disease, tau protein can become hyperphosphorylated, which just means it has too many phosphate groups added to it. And this can mean it can aggregate into insoluble structures known as these neurofibrillary tangles. And tau aggregates, which is those clumps, they can disrupt normal cellular function, including that axonal transport. And a big reason why it disrupts certain processes is once these clumps form, they tend to migrate away from the axon, relocating in dendritic spines, where they can alter certain processes such as postsynaptic glutamate receptor trafficking, which is a bit of a mouthful, or anchoring, so that process by which receptors are integrated into these postsynaptic membranes, such as glutamate receptors, and thus this can suppress excitatory postsynaptic currents and neuronal activity. These do. I'll just explain. This is quite a hard concept if you're quite new to neuroscience, but they're very important when looking at functions of interneurons, and these play a role in generating oscillatory, being the types of brainwave activity that contributes to normal cognitive function. Within the brain, you have these oscillations and waves, and a wave is defined as it's a group firing of neurons in specific patterns, is really what a wave is. And as you can imagine, these tau proteins can disrupt that. So when we move on to amyloid beta is basically a metabolite. It is originally something called amyloid precursor protein, or APP for short. And this amyloid precursor protein is used in many processes, such as cell growth, cell signaling, and synapse formation. And it can be broken down by two pathways, and there's actually a couple more, but one of these pathways produces the amyloid beta, which has a tendency to clump together and form insoluble oligomers and fibrils. And these can eventually become these plaques that are involved in disease. And the disease state is caused by many things, but one of those is the reduced astrocyte glutamate uptake. So if you can cast your mind back even earlier in the video, and we were talking about astrocytes and how important they are, one of their jobs is to reuptake excess glutamate and repurpose it and recycle it into either more glutamate or GABA, but essentially broken down, taken out of that synapse, and then it can be repackaged in the presynaptic or postsynaptic membrane. And if you look at the diagram, we have a synapse down here in the bottom left hand corner with your presynaptic membrane, your postsynaptic membrane. This purple structure monitoring what's going on in the synapse is your astrocyte. And without it, or with its reduced function due to those amyloid beta plaques, you're going to get excess extracellular glutamate, and this is going to increase neuronal excitability. If you think back to glutamate being the main excitatory neurotransmitter in the central nervous system. And if you have too much firing, this is going to alter synaptic plasticity, and in extreme case scenarios, you're going to get a bit of exotoxicity. But I think this diagram's really interesting, looking at a bit of epigenetics and these tangles in the form of going from a healthy brain to a dementia or a brain within a disease state. And I just wanted to outline those two waste products because they're heavy hitters or big topics within research at the moment, and I kept mentioning waste products, but I never defined any, so I hope you found this useful. In terms of movement, you're also looking at those HDLs, so I mentioned earlier the movement of cholesterol around the brain, and then also things like insulin. Insulin it's gaining more traction and we're knowing more about it, but insulin has major roles within the brain. So now we're going to move on to sleep. So sleep. If you ever search on PubMed for articles on the glymphatic system, you will instantly see papers looking at the glymphatic system and sleep. And much of the information, even for this video, comes from an article titled Sleep, Cerebral Spinal Fluid and the Glymphatic System, a Systematic Review. And research has shown that the glymphatic system is most active during sleep. And although it's constantly filtering these toxins from the brain, during wakefulness the system remains mainly disengaged. And although sleep is often associated with rest, this glymphatic system activity is dramatically more active during sleep. And photoimaging of in vivo mice demonstrated a 90% reduction in glymphatic clearance during wakefulness, which is crazy, as a comparison, and even twice the amount of protein clearance from the brain happens during sleep compared to wakefulness. And then, pardon me again, A lot of this is often attributed to that expansion of interstitial space, which can increase from like 13-15% all the way to 22 to 25% increases during sleep. And it's been looked at during using fMRIs and EEGs in order to detect which type of sleep most of this clearance and toxin removal comes from, and it's quite apparent that it's mainly during deep sleep. So within sleep you have many types of sleep. So you've got your REM, which is rapid eye movement sleep, and then you've got non-REM, and then non-REM can be divided into another 3 stages, and it would be non-REM stage 3 being your deepest sleep, where the main cleansing of the brain using that glymphatic system is observed. And it's showing these slow oscillations of, and these brain waves also increase the amount of CSF within these interstitial cavities, leading to that 90% increase in glymphatic system, with the glymphatic system even. Interestingly, when you think about the mechanisms triggering sleep, you think of that circadian rhythm of your suprachiasmatic nucleus, talked about by Matthew Walker, and many people in that sleep-wake pattern that we follow roughly over that 24-hour period. However, when initially looking at this phenomenon, scientists did so using anaesthetized mice, which is an artificial sleep, which, as you can tell, may seem very inaccurate as regards to mimicking sleep, because it's definitely not the same. Sleep is more complex than just a sedated state. However, the sedated state had almost identical results to later on studies that looked at sleeping mice compared to these originally anaesthetized mice, showing that the glymphatic system working was not simply to do with that circadian rhythm. And so it's hypothesized that it's more to do with noradrenaline. Noradrenaline is a neurotransmitter responsible for concentration and alertness during awake states. And it may also be to do with insulin, might have a role in the glymphatic system activation. But we do know that it's actually not as much to do with noradrenaline, which is all the more reason to take things like sleep apnea and even snoring, which can be a sign of sleep apnea, more seriously. Like, it is something to actually look at and pay attention to, especially if you notice this in friends or family. It's worth mentioning and having a look at. So if I go through my diagram now that I've made using Biorender before they sue me for not giving them credit, we've covered that increase in interstitial space. We've got that increase in metabolic clearance and waste products, facilitated by that increase in cerebral spinal fluid flow. Remember, we're making that 500 milliliters a day roughly, but we can only hold that roughly around 150 milliliters a day, so it's constantly being pushed through and flushed, and this is more apparent when we sleep. The glymphatic system being more active due to this. It's more pronounced and during slow wave frequencies, such as non-REM sleep, stage 3, that really deep sleep that we have, is when the glymphatic system is most active. And one of the key regulators hypothesized for the activation of the glymphatic system is noradrenaline, rather than things that regulate that circadian rhythm. Okay, let's move on. As with many biological systems, age is not kind. And the glymphatic system is no exception to this rule. Some things that run as predictors or biomarkers for early dementia often overlap with dysregulation of the glymphatic system. So as we get older, we have a decrease in that cerebral spinal fluid production, and then this leads to a decrease in cerebral spinal fluid pressure. And then on top of this, we also have a decrease in those vital aquaporin 4 channels that we mentioned earlier. And this is all relating to the dysregulation of the astroglial water transport system that helps make up our glymphatic system. So that all sounds a bit morbid and not too hopeful for the future. But very interestingly, the glymphatic system also shows one of the key pathways by which head trauma, which I mentioned earlier, even in multiple and single events, inflicts damage, as head trauma has been shown to decrease the action of the glymphatic system for up to 28 days, which is crazy, a month. Even a month post that first impact or multiple impacts, your brain's still trying to recover. And along with a decreased aquaporin 4 action, your brain can develop astroglial scars within the brain tissue, and this can be viewed using imaging methods. And this is also coinciding with an increase in amyloid beta and tau due to that less active glymphatic system. And altogether, this is what you would call a recipe for disaster. So I just wanted to mention that. Those are really just the key points. So when we look at age, increased age, we have that decreased CSF production, which leads to a decreased CSF pressure. Compounding with this, we have a decrease in aquaporin 4 channels and function. So this can contribute to less clearance and more buildup of these waste products that we don't want, leading to a dysregulation of the system, which can lead to many of the almost side effects, if you will, and unhappy symptoms that we see in more elderly people, such as dementias, can be as a result of a decrease in function of this system. And then if we recap it again, that trauma can also, so with trauma, you're going to have an increase in these metabolic waste products to begin with, but you're also going to have a decrease in clearance. And this can lead to things such as glial scars and that decreased clearance within the system. So this is not a good situation. This can be why concussions and head impacts can be all the more serious than you would believe, as the damage can last up to months at a time to recover from them. And if you think people playing rugby matches or hockey matches, these can be once a week. So you may start feeling better by the next week by the time you play a next game, but you might not be recovered to a month later, and then you're taking another impact, and that's delaying your recovery even further. And it may, if you play the whole winter, it may take you all of those summer months back up to your next season, and even then you might not be recovered. And over years this can compound and lead to a lot of nasty conditions that is sad to see in many ex-professional contact sports players. So I just want to say thank you for watching. I hope you have found this especially interesting, as it's able to tie in so many previously learnt topics and gives them a bit more weight, a bit more relevance in the growing world of research. So anyway, goodbye.